Welcome back. We're almost finished with this section. We're going to talk about random fertilization, and then we're going to briefly compare some mitosis with meiosis. So first, what is random fertilization? Well, a zygote is when we have the random union of those gametes which were produced independent of each other. So we had a sperm that was haploid produced in the male, and we had an oocyte that was haploid produced in the female, and their productions had nothing to do with each other. And when the sperm fertilized the oocyte, that creates what's called a zygote, and that zygote is now diploid. Okay. And random fertilization adds to our genetic variation. Remember, it's the third thing that's adding to genetic variation. We had one thing that added in prophase one. We had something else that added in metaphase one. And now here is our third thing that happens. And the reason why this is considered adding genetic variation is because any sperm can fuse with any ovum. Okay, there's no rules to it. So how do we determine what sperm fertilizes with what egg? What are the odds? How do we know what our choices are? Well, remember there were almost 8.4 million possible combinations for each sperm and 8.4 million combinations for each oocyte. And so we take that 8.4 million and we multiply by another 8.4 million. That means there are over 70 trillion possible combinations to form one of you. And that's not even including any variability from crossing over in prophase one. So now do you understand why you don't look like your siblings? Why you don't have the same likes and dislikes? Yeah, it's mind-boggling sometimes, isn't it? All right, so let's go back and hit some details comparing mitosis versus meiosis. Let's take it down to the very, very basics, okay? Because when you're learning science, you have to learn the basics before you can add on the details. And oftentimes students focus on all the details, like when they're looking at a forest, they're focusing on, well, the leaf looks like this and the flower looks like this and the seed looks like this, rather than recognizing it as a forest or this type of tree or that type of flower. So in mitosis, we started with one duplication and interphase, and then in mitosis, we did one division. Comparing that to meiosis, where we had one duplication and interphase, and then we had two divisions. Okay. In mitosis, at the end of the one division, we had two daughter cells. Each daughter cell was identical to the parent, and each daughter cell was identical to each other. In meiosis, we ended up with four daughter cells. None of the four daughter cells were identical to each other, and none of the four daughter cells was identical to the parent. Meiosis also had crossing over, and we could see crossing over evidence in the daughter cells. And we also had independent assortment, and we could see evidence of that as well. So we start with diploid 2N cells. We end up with haploid N cells. Okay, so here's your review with pictures of mitosis, the blue background, and meiosis with the green background, with both meiosis 1 and then going to meiosis 2. Give at least 3 facts how meiosis was different from mitosis. Walk through the process of meiosis 1, the process of meiosis 2, then go back and think through mitosis and figure out, is meiosis 1 more similar to mitosis, or was meiosis 2 more similar to mitosis, and why? Do some critical thinking. That's it for this. And we're going to come back and talk one more time because sometimes there's some oopsies that occur. And we're going to see what happens when there's an oopsie. Thank you, and I'll see you shortly.